Hello everyone, I'm MSLs, and this is session 41 or lecture 41 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug series and drug chain interaction trials. Introduction. Definitions: Series interaction trials assess the sequential effects of administering multiple drugs in a series, examining how one drug's impact modifies the outcomes or efficacy of subsequent drugs in the sequence. Drug chain interaction trials focus on the interconnectivity of different drugs taken concurrently or in a chain, analyzing cumulative effects, interactions, and potential synergies or toxicities. Key points. Drug series interaction definition. Again, drug series interaction trial refers to the effects that emerge when drugs are administered sequentially. This interaction can influence the pharmacokinetics (PK) or pharmacodynamics (PD) of subsequent drugs in ways that may enhance or impair therapeutic efficacy. And here's a summarized diagram on understanding drug series interactions, mitigating sequential toxicities, enhancing efficacy, metabolism, and bioavailability. Key focus areas in drug series interaction trials, we have enhancing efficacy through sequencing. Some regimens are structured to maximize efficacy through carefully timed sequences. Example, pre-treatment with corticosteroids before chemotherapy reduces inflammatory responses, making the chemotherapy more tolerable and effective. Metabolism and bioavailability, the first drug in a series can impact the metabolic pathway of the following drug, altering absorption rates or bioavailability. Example, a hypertension regimen administered beta blockers, drug 1, before ACE inhibitors, drug 2. However, the delayed effect of drug 1 metabolism caused amplified side effects from drug 2. Mitigating sequential toxicities, cumulative toxic effects must be carefully monitored in sequential regimens. Dosages may need adjustment to prevent cascading adverse effects. Example, antibiotics in tuberculosis TB treatment are administered in a sequence to minimize resistance and toxicity, with drug 1 paving the way for more efficacious action by drug 2. Drug chain interaction. Definition. Drug chain interaction trial examines the concurrent use of multiple medications. Assessing these interactions is critical in understanding cumulative or synergistic effects, as well as identifying potential toxicities from overlapping mechanisms of action. Here's a summarized diagram summarizing the understanding of the drug chain interactions. We have on the left synergistic effects, on the right overlapping side effects, in common area in between enhanced adverse outcomes. Key focus areas for drug chain interaction trials: synergistic effects. When drugs interact positively, they can enhance therapeutic outcomes beyond what either would achieve alone. Example, combination HIV therapies involving integrase inhibitors and protease inhibitors leverage synergistic viral suppression mechanisms to significantly improve patient outcomes. Overlapping side effects. Drugs with similar side effect profiles can amplify adverse events when taken simultaneously. Example, heart failure patients taking ACE inhibitors and diuretics may experience severe hypotension due to additive effects on blood pressure reduction. Pharmacokinetic and pharmacodynamic conflicts. Concurrent drugs may compete for the same metabolic enzymes, reducing efficacy or increasing toxicity. As for example, a cancer regimen combining two chemotherapeutic agents revealed that one drug inhibited the metabolism of the other, leading to toxic plasma concentrations. Key components of monitoring: patient safety or participant safety. Focus prevent adverse interactions and cascading toxicities in both drug series and drug chain regimens. Actions regular monitoring for cumulative toxicities such as liver or kidney dysfunction. Close observation of high-risk populations, including pediatric, elderly, or renal compromised patients. Example, a cardiac regimen monitored hypertension risks when combining beta blockers and ACE inhibitors. So a cardiac regimen monitored hypotension risks when combining beta blockers and ACE inhibitors. Um, Here's a summarized diagram on ensuring patient safety in healthcare. On the left, patient safety. On the right, prevent adverse interactions, monitor cumulative toxicities, and observe high-risk populations. Data integrity. After patient safety and key components of monitoring, and as in data integrity, the focus ensure accurate and consistent data collection on PK, pharmacokinetic, PD, pharmacodynamic parameters for all drugs involved, actions, standardizing tools for measuring drug interactivity parameters. First, site validation to ensure uniformity in data. Example, on HIV trial validated drug absorption consistencies by analyzing differential plasma levels for the first drug, delaying the activation of the second. Protocol adherence focus here strict adherence to timing, sequencing, and combination protocols. Actions train research staff on standardizing administration procedures, accurately document any deviations that could introduce bias in results. Example, a diabetes trial maintains strict timing for insulin injections followed by oral medications to ensure efficacy optimization. The regulatory compliance focus here adhere to legal and ethical standards that govern multi-drug regimens. Actions submit comprehensive safety evaluations on drug regimen interactions, ensure transparency in documenting adverse outcomes. Example, a cancer trial submitted detailed safety profiles for a pre-treatment regimen involving anti-inflammatory drugs combined with immunotherapy. Steps to effective monitoring. And here, before we proceed with the steps to effective monitoring, as here we have the diagram summarizing the effective monitoring process. On the left side selection, we have risk-based monitoring here, data review, and then enhanced patient safety. So step one is site selection. Sites must have expertise in multi-drug therapy administration and advanced monitoring technologies. Example, pediatric antibiotic trial selected hospitals with in-house renal monitoring capabilities to manage potential toxicities. Step two would be, as here in the diagram, risk-based monitoring, RBM. Focus on subpopulations at higher risk of adverse interactions. Example, elderly patients with comorbidities received increased monitoring for cumulative toxicity from sequential hypertensive medications. Step 3, data review. Regularly evaluate collected data for inter-drug anomalies or trends. Example, a TB, a tuberculosis regimen flagged reduced efficacy for drug 2 due to upstream induction of metabolic enzymes by drug 1. We have tools and technologies for monitoring. We have here pharmacokinetic analysis tools here for pharmacokinetic analysis, track drug metabolism and interaction dynamics. Example, tools assist delayed absorption patterns caused by a pre-treatment regimen during bacterial infection trials. Simulation and predictive models model in vivo interactions and preemptively identify risks. Example, models predicted gastrointestinal GI toxicity in cancer regimens involving sequential chemotherapies. Centralized data platforms offer real-time analysis of multi-drug regimens across research sites. Example, centralized monitoring identified cardiac complications caused by a hypertension drug series. Summary tables. Next, we have tables to summarize what we have covered through this lecture. As here we have a table on key components of monitoring, as we have the component, the focus, the actions, example, outcome. Components, we have patient safety, data integrity, protocol adherence, regulatory compliance, monitoring efficacy. Here we have table on steps to effective monitoring, step focus, key actions, example, observed outcome. And steps here we have site selection, risk-based monitoring, data review, protocol alignment, technology use. Table on tools and techniques for monitoring. Tool technique, purpose, feature, advantage, example, implementation. As tools techniques, we have EDC, electronic data capture, pharmacokinetic analysis, PK analysis, simulation modeling, centralized monitoring, real-time biomarker tools. Table on examples of successful monitoring. Trial application, drug type, outcome focus, key findings, patient impact. Trial applications, we have antibiotic sequences, cancer immunotherapy, pain management systems, heart failure protocols, HIV multi-drug therapies. Application examples. Scenario, drug example, focus approach, impact on healthcare. Scenarios, we have pre-surgical anti-inflammatory use, sequential cardiovascular management, chronic pain dual therapy, HIV multi-drug regimens, antibiotic resistance management. Application table, scenario for each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios, we have testing the impact of pretreatment steroids on chemotherapy efficacy, monitoring sequential beta blockers and AC inhibitors in cardiac patients, evaluating drug metabolism shifts in an HIV regimen due to sequential combinations, using EDC, electronic data capture systems, to track adverse effects in pediatric multi-drug antibiotic regimens, optimizing NSAID opioid sequences for post-operative pediatric pain, simulating gastrointestinal GI toxicities in combined chemotherapy regimens, monitoring potassium shifts during diuretic and ACE inhibitor sequences for heart failure patients, evaluating timing impacts on diabetes injections followed by oral medications, monitoring therapeutic duplication effects in hypertension combination regimens, investigating delayed side effects when layering steroids with immunotherapies in arthritis patients, monitoring renal outcomes from antibiotic interactions in pediatric nephrology, identifying enzyme inhibition issues in delayed absorption cases for sequential TB drugs, tuberculosis drugs, adjusting doses of ACE inhibitors followed by diuretics in hypertensive patients. And here we have assessing efficacy of multi-drug regimens with centralized real-time monitoring. Next, we will revise what we have covered through this lecture through some MCQs, multiple choice questions. Question one, what is the focus of drug series interaction trials? A, the development of entirely new pharmaceuticals. B, assessing the sequential effects of administering multiple drugs. Monitoring economic impacts of pharmaceutical production. C, and or D, testing drug efficacy in single dose regimens exclusively. So the answer is B, assessing the sequential effects of administering multiple drugs. As drug series interaction trials evaluate how administering multiple drugs in succession affects outcomes, such as whether one drug influences the efficacy or safety of drugs in the sequence. Subsequent drugs in the sequence. Question two, why is patient safety critical in drug chain interaction trials? And here, patient safety is critical in any kind of trial, not only in drug chain interaction trials, with all trials. So, A, to ensure multi-drug regimens do not produce cascading toxicities or cumulative side effects. B, to avoid scheduling effects when administering drugs. C, to focus exclusively on the elimination of the regulatory hurdles. Or D, to negate the need for centralized data systems. So definitely here the answer would be A, to ensure multi-drug regimens do not produce cascading toxicities or cumulative side effects, as patient safety ensures adverse effects from drug interactions within a chain, such as overlapping side effects, are monitored and mitigated effectively to protect participants. Question three, which tool is essential for tracking drug administration in multi-drug trials? Is it A, pharmacokinetic PK analysis tools, B, manual data entry logs, C, electronic data capture EDC systems, or D, simulation frameworks for single dose regimens? So answer is C, EDC systems is a tool that is essential for tracking drug administration in multi-drug trials, as EDC systems allows accurate scheduling and tracking of sequential or chain drug administration while recording side effects and outcomes in real time. Question four, what is a common challenge faced in conducting drug series interaction trials? A, delayed peak plasma levels due to enzyme inhibition from previous drugs. B, frequent changes in trial endpoints. C, lack of transparency in patient recruitment processes, or D, screening for pharmacodynamic effects, PD effects in placebo-only trials. So what is a common challenge faced in conducting drug series interaction trials would be A, delayed peak plasma levels due to enzyme inhibition from previous drugs or previous medications, as delayed drug efficacy due to enzyme inhibition by a previously administered drug in the series exemplifies a key pharmacokinetic PK challenge in these trials. Question 5. How do simulation and modeling benefit drug chain interaction trials? A. By reducing the need for regulatory compliance altogether. B. By predicting potential drug interactions before clinical trials begin. C. By avoiding the necessity for PK, pharmacokinetic analysis tools. Or D. By replacing real-time data capture with theoretical models. So how do simulation modeling benefit drug chain interaction trials? As the answer is B, by predicting potential drug interactions prior to beginning the clinical trials or before clinical trials begin. So answer would be B, as simulation and modeling help foresee potential interactions or cumulative toxicities between drugs, allowing researchers to design safer and more efficient drug chain protocols. And here we come to the end of this lecture. Thank you very much and goodbye.